All right. Moving on, chapter 5, microbial metabolism. This is again going to be a long chapter. I promise there are other short chapters. Compensate. Okay. I'm sorry. But microbial metabolism is really important concept to understand. And the reason why it's important to understand is because if you understand microbial metabolism, you understand how to control it. And therefore, one of the things, I mean, you guys are going to be nurses, okay? And one of the things I really want you to understand is not like all the intimate details about different microbes. It's really how you guys can control it and how we as a society, especially in the medical field, control microbes. And one of the ways we do that is understanding the metabolism of different microbes and figuring out ways to prevent that from happening. Okay, no metabolism, microbes are either inert or dead. Okay, all right, hopefully we can get to, okay, so. Okay, metabolism is the buildup and breakdown of nutrients within a cell. So all the chemical reactions that happen inside a cell either provide energy or create the substances that sustain life. Okay, so they either provide energy to build stuff or they build stuff. Okay. All right, so big picture, metabolism is broken down into two parts, anabolism and catabolism. Anabolism is building up of stuff, catabolism is the breaking down of stuff. Okay, so basically what happens inside a cell is both. So I take stuff that I don't no longer need or I eat, and I break it down into individual components. Those individual components I use to assemble what I want. Okay, it's like Legos, right? I take my Legos I built yesterday and I break it down into little pieces again, and then I rebuild what I want today. Okay, and so that is metabolism. Okay, although microbial metabolism causes disease, okay, and food spoilage, many ways are beneficial rather than pathogenic. In fact, we abuse microbes in ways to make stuff for us. We hijack the metabolism to make stuff for us. Okay, like alcohol and bread. I said alcohol first, but anyways, all right. So define metabolism. Okay, so we talked about this already. Okay, ATP is this thing in the middle. Okay, ATP is what goes in between metabolism and catabolism, because when we break down stuff, we get energy from breaking down stuff. We take that and we store it as ATP. When microbes build up stuff, they have to use energy, and that energy comes from ATP. Okay, so hopefully that makes sense. Okay, so metabolic pathways, we'll talk a little bit about pathways in a little bit, but metabolic pathways are sequences of enzymatically catalyzed chemical reactions within a cell. What that means is you never go from A to Z in one single step. You go from A to B, okay? That A to B is encoded by an enzyme. That enzyme is encoded by a gene, okay? You go from B to C, that B to C is controlled by another enzyme, and that enzyme is controlled by another gene, okay? C to D, another enzyme, another gene, okay? All the way to the very end, okay? And yes, even more trickier than that because they branch and other stuff, but you know, we like to think of metabolic pathways as a series of steps, okay? All right. Let me stop sharing. Second. let me enable the computer sounds and stuff like that. So. Okay.
As organisms live and grow, they're constantly in the process of making and breaking bonds in molecules. Metabolism is the sum of all the chemical reactions that take place in an organism. Metabolism includes catabolism and anabolism. Catabolism includes the processes that break down complex molecules into simpler molecules while harvesting their energy and storing it, usually in the form of ATP. Anabolism includes the processes that build more complex molecules from simpler molecules. The energy acquired through catabolic processes is used to drive anabolic processes. Neither catabolism nor anabolism is completely efficient, so at each step, some of the available energy is lost into the environment as heat.
All right. Okay. So enzymes, okay? Learn the components of an enzyme, the mechanisms of enzymatic action. Okay. So you don't have to understand collision theory, okay, but you have to understand activation energy. Activation energy is the energy that's required for a chemical reaction to occur. So for a chemical reaction to actually occur, these two things, okay, these two chemicals have to have high enough energy and they have to actually run into each other, right? Chemical reactions are not going to magically occur across the room. They're going to only occur when two things actually come in contact. And not only do they have to come in contact, they have to come in contact at high speed, okay? Collide, okay? Okay, the reaction rate is the rate at which chemical reactions happen, okay? Chemical reaction rates can be increased by enzymes or by increasing temperature, okay? If I increase temperature, things move faster and they collide more often because they move faster. I increase pressure, I push these things together more, and so more reactions happen. Concentration, the more of these things floating around, the more collisions will happen. And okay. All right. So enzymes. Okay. So let's see what enzymes actually are.
Imagine a chemical reaction with the general form AB yields A plus B. In order for this reaction to occur, the reactant molecule, AB, must acquire enough energy to disrupt its stable electron configuration so that its electrons can rearrange to form A and B. This energy barrier is called the activation energy of the reaction.
Yeah. So make sure you understand that. Okay. So
in living cells, enzymes serve as biological catalysts, speeding up reactions. Enzymes accomplish this task by lowering the activation energy of the reaction, making it easier for reactants to acquire the activation energy required to produce products.
Okay, so what I'm doing here, okay, is an enzyme is lowering the activation energy, the amount of energy necessary for the enzyme for the chemical reaction to work. Okay, so if you look at 1,000 people, those 1,000 people have different amounts of energy. Some people are really energetic, and some people can't get out of bed. Okay, but so on average, people have a certain amount of energy. So what happens with enzymes is the same thing. These molecules all have different amounts of energy. But if you have a lower barrier to activation, then more molecules can cross over that barrier, and therefore the reaction will happen faster. Okay? Okay. All right. So what actually happens with enzymes? Let me show you. Okay. So the blue blob is the enzyme. the red blob is the substrate. Okay, so mostly the enzyme. Okay, in reality, the big blob, the blue blob, is much bigger than the red blob. Okay, but they do literally look like blobs. Okay, the little site there is called the active site. Okay, for the enzyme fits the substrate onto its active site, and then the enzyme substrate complex, once that's been established, it changes shape a little bit to where it makes it really easy for the chemical reaction to happen. Like if you're breaking something, that bend like stretches out the bond so they break easier. If they're putting things together, the bend puts those things physically very close to one another so the chemical reaction can happen easier. Okay? But basically the enzyme substrate complex has to change after the attachment of the substrate to the enzyme. And that change is allow the substrate to undergo whatever chemical reaction is supposed to easier. And that is the mechanism of enzyme action. Okay? So, okay. After the enzyme does this thing, the substrate falls off the enzyme, and now it's called product. Okay, product is released. Okay, the enzyme can do it all over again because it's not actually used up. It catalyzes the reaction, and it can do it again. Okay. So watch the video.
Although every enzyme performs a different reaction on different substrates, they all go through the same general steps in accomplishing their reaction. These general steps include substrate binding, formation of the enzyme substrate complex, product formation and dissociation, and enzyme recovery. Like all catalysts, enzymes are unchanged by the overall reaction. Now let's look at each step in more detail. The first step in any enzymatic reaction is the binding of the substrate by the enzyme. The enzyme includes an active site that can accommodate the size, shape, and electron configuration of its substrate or substrates. According to the induced fit model of enzyme-substrate interaction, a perfect fit does not form until the enzyme and substrate bind to form an enzyme-substrate complex. After the substrate is bound...
All right, I'm gonna stop that right there. We'll talk about induced fit. Induced fit, imagine two people shaking hands. As their hands come closer to each other, their hands have to change shape, okay, for one person's hand to fit on the other. If everybody stuck their hands out like this, like you wouldn't be able to shake hands like this. You would only be able to shake hands if I grabbed around your hand and you grabbed around mine. Okay, that is an induced fit model. Okay.
In the active site, the enzyme substrate complex is formed. The shape of the enzyme may change to allow for an efficient chemical reaction, and amino acids may accept or donate protons or electrons to facilitate the chemical reaction. Once the enzyme substrate complex is formed and any reactive groups have moved to the right position, the reaction proceeds and products are formed. The products dissociate rapidly, leaving an empty active site. After the products leave the active site, the enzyme returns to its original configuration. Any groups that donated or accepted protons or electrons are also returned to their original state. Like all catalysts, the enzyme is unchanged by the overall reaction.
Okay, so there you go. So you can see there that there's a slight change in the enzyme as the substrate enters, right? So, okay, enzymes have specificity for specific substrates, okay? So not one enzyme does work on everything. Enzymes work on very specific things. The substrate has to be able to fit inside that hole and fit properly. Okay. Turnover rate is how fast the enzyme actually works. Okay, and it usually works pretty damn fast. Like the one, per 1 per second is really low. It's more like 10,000 per second. Okay, it can go as high as 500,000 per second. Okay. So how enzymes are named. So these are how enzymes are named. Okay, so they're named pretty logically. Oxidoreductase, oxidation reduction, transferase. Okay, so first of all enzymes end in ASE. The only exception to that rule are really old enzymes that we know about that that was discovered before this rule was invented. Okay, but basically all of these things you can see like they're named after what they do. Okay, isomerase makes isomers, right? Rearrangement of atoms are called isomers. Okay, lysase remove things. Okay, hydrolysis, hydrolase. Okay, ligase ligates things together. Okay. Okay. Don't worry about the components. Okay, enzyme and don't worry about apo enzyme or hollow enzyme, but know what a cofactor and a coenzyme is. A cofactor is a non-protein component that makes the chemical reaction better or faster or like improve the enzyme somehow. Okay, a coenzyme is a organic cofactor. This is what we actually vitamins are. Minerals are inorganic cofactors. That's why we need minerals as well. Okay. Okay. Enzyme. These are electron carriers. Okay. NAD, NADP, FAD, coenzyme A. These things are going to carry electrons, and you'll find out very shortly why that's important. Okay. All right. Factors influencing enzyme activity. Okay. Again, I already went through this. Temperature, pH. Okay. Substrate. So temperature. The faster temperature is, the higher, the faster the enzyme activity rate up to a certain level. Okay, too high at temperature, and you can actually denature the enzyme and it will no longer work. Okay, pH. Okay, again, you need to be at the right pH for that enzyme to work, and the right pH depends on what enzyme it is. Okay, the substrate concentration, increasing the substrate concentration will always make the enzyme work faster, up to a certain amount, and then it just maxes out as fast as it can, and it doesn't matter if you increase substrate after that. Okay, enzyme inhibitors are things that purposely inhibit the enzyme, and we'll talk about what they are in a little bit. Okay. Okay. So this is denaturation of a protein. This is what happens when you add too much acid, too much base, or too much temperature to a protein. It denatures, it unravels. If it unravels a little bit, going back to normal will make it fold back. But if you denature it this far, you can change it back to normal and it won't refold again. Okay. Okay. Enzymes only work at whatever temperature they're designed to work at. Okay, our body is 37 degrees, so our enzymes work best around 37 degrees. Notice that after 40 degrees, and even a little bit before 40 degrees, the enzyme activity rate goes down. Okay, so when you're spiking a really high fever and stuff, not particularly good for you. Okay, the only benefit is that it's worse for the bacteria. Okay, and same for pH. this is substrate concentration. So when I add enough substrate, all the like, so if I give you like one assignment, okay, so you can do it rather quickly. If I give you 2 assignments, you'll be motivated to work a little bit faster. But there comes a time when I give you enough assignments and you're literally working as fast as you possibly can. You're not sleeping, you're not eating, you're not playing, you're just working as hard as you can. But working as hard as you can only get so much done. And the more assignments I pile on, it doesn't change anything. Okay? All right. Inhibitors. Okay. Inhibitors, there are 2 types of inhibitors of enzymes. One is called a competitive inhibitor, and one is called a non-competitive inhibitor. Make sure you understand the differences between the 2. Okay. So this is a competitive inhibitor. A competitive inhibitor competes for the active site with the substrate. Usually they bind the substrate at a much tighter level than the substrate does. So the competitor outcompetes the substrate for the active site. Okay. Okay.
A competitive inhibitor is a substance that is structurally similar to the enzyme substrate, but which does not react to form products. For example, sulfanilamide is a competitive inhibitor for the enzyme whose normal substrate is para-aminobenzoic acid, PABA. Note the similarity in molecular structure of PABA and sulfanilamide. In competitive inhibition, competitive inhibitors compete with the substrate molecules for the binding site, slowing catalysis. However, if the concentration of substrate is much higher than the concentration of competitive inhibitor, the substrate will outcompete the inhibitor for the enzyme active site, and catalysis will continue.
Okay. Okay, so for a competitive inhibitor, you can outcompete it. So to outcompete it, all you gotta do is add more substrate. Okay, so if I add for a competitive inhibition, all I have to do is add more substrate to overcome it. Okay. Non-competitive inhibitors attach to a second site called an allosteric site. And that process is called allosteric inhibition. Okay, by attaching to that second site, I change the active site so the substrate can no longer bind. And by doing that, it doesn't matter how much substrate or how little substrate I have, it just won't work. No matter how much more adding more substrate won't fix the problem. Okay, so here is the non-competitive inhibitor site. It is not the active site. Okay.
Non-competitive inhibitors bind to a location on the enzyme other than the active site. For example, the HIV medication nevirapine is a non-competitive inhibitor of HIV reverse transcriptase. Nevirapine does not bind to the reverse transcriptase active site. The binding of the inhibitor causes the enzyme to change shape, so the active site is no longer able to perform the reaction efficiently. Since the non-competitive inhibitor binds in a different binding site than the substrate, increasing the concentration of substrate has no effect on non-competitive inhibition.
Okay, make sure you understand that difference between competitive inhibition and non-competitive inhibition. Okay, feedback inhibition is always a non-competitive inhibition. Okay, so usually, okay, you have a substrate, so this substrate activates. You have end product. The end product will bind to enzyme number 1 usually in an allosteric site and cause non-competitive inhibition. So the only way I get this system going again is not by adding substrate. It doesn't matter how much substrate I have to get rid of the end product. If I get rid of the end product, if I use up all the end product, then I can go through this pathway again, okay? Feedback inhibition. So once I run out of products, I make more products. If I have too much products, I stop making products. Okay, feedback inhibition. All right, ribozymes. Ribozymes are organic catalysts as well, but they're not made out of protein. They're actually made out of RNA, and RNA catalyzes the reaction. Okay, they're frequently used to cut up and splice RNA. The enzyme that does that has a really cool name called dicer. And then they're also involved in protein synthesis in ribosomes. The ribosome is more accurately described as a ribozyme, okay, with protein components to it. Okay. All right. Okay. So explain the term oxidation reduction. So I'm sure you guys have heard this term before, but I'm gonna try to explain it in a clearer way or in a more biological way that's easier for you guys to understand. For you guys that don't like AP chemistry and stuff, you can delete that from your brains. Oxidation reduction in biology has much more specific terminology to it. Okay. The easy way to remember this, okay, is oil break. Okay, oxidation is loss, reduction is gain of electrons. Okay, now that being said, what's more important is what the electrons represent. The electrons represent energy. If I add electrons, I'm gaining energy. Okay, so oxidation is the loss of energy, reduction is the gain of energy. Okay, so every time I add electrons to a organic thing, that thing gains more energy. Every time I remove electrons, I lose energy. So when I oxidize something, that thing loses energy. Okay. Okay. And usually what we measure is the amount of hydrogens. Because hydrogen comes with electrons. Okay. So if I have an increase in hydrogen levels, okay, I am reducing. If I lose hydrogens, I'm oxidizing. Okay, because where the electron goes, the hydrogen goes with it. Okay. All right, so electrons, so B is reduced and then A is oxidized. So in oxidation reduction reactions, one thing gets reduced, one thing gets oxidized. A is oxidized, B is reduced. Okay, the thing that is an electron donor is oxidized, the electron acceptor is reduced.
Many metabolic reactions involve the transfer of electrons from one molecule to another. This transfer of electrons involves two coupled reactions, oxidation and reduction. These are often called redox reactions. Cells use redox reactions to transfer electrons from one molecule to another within or between metabolic pathways, such as the Krebs cycle and electron transport chain. In an oxidation reaction, a molecule called the electron donor gives up one or more electrons to another molecule called the electron acceptor. In the oxidation reaction, the donor molecule becomes oxidized. A reduction reaction occurs when a molecule receives electrons from another molecule. The electron receptor therefore becomes reduced. The term reduced refers to the receptor's electrical charge. Because it's gained a negatively charged electron, the overall charge on this molecule becomes more negative and is said to be reduced. You can now see that the oxidation reaction must be coupled with a reduction reaction. In other words, if a molecule is to donate an electron and become oxidized, then another molecule must accept that electron and become reduced.
Okay. So in biological systems, electrons and protons are removed at the same time. So it's equivalent to a hydrogen atom. So the biological oxidations are often dehydrations. The biological reductions are usually hydration. So you add hydrogen. Okay. Okay. So remember, I explained when we went over the assisting enzymes and stuff. I said that NAD was important because, and I kind of left it. I'll explain why that's important. This is why. NAD plus is a very effective electron carrier. So this is the oxidized form. Okay, NADH is the reduced form. The reduced form can carry a lot of energy. Okay, and we can use the NADH to carry energy to other places, and we do. And you'll see that in the next chapter, okay, or next recording, okay? All right.